All right, so we'll move on to our next speaker. So our next speaker is Katherine Moir from Queen's University, and she'll be talking about diatom responses to industrial contamination on the St. Lawrence River. So I'm going to share your presentation. You'll be here at view control. There you go.
Okay, great. Thanks, Christina. So as she said, I am a PhD student at Queen's in the Department of Biology, where I'm in the Pearl lab. We have another person from our lab, Zoe Armstrong, presenting right after me. And so we're going to both be talking about some of the implications of legacy sedimentary contamination on the river at Cornwall. I think it's thinking. Hold on. Maybe. Come on. There we go. So probably a lot of people on this call are well versed in kind of the history of the area, but if you're not, we're located on the river, St. Lawrence River at Cornwall. This region has seen a lot of industrial activity through much of the 20th century on both the north and south shores of the river. This talk's going to be focused just on the Canadian side, on the north side. And throughout much of this history, especially in the early days of industrial activity, there was a lot of effluent that was diverted directly into the river before, pollution was really much of a concern as an issue, before regulations went in limiting controls on what could be in your effluent. And we could see that in the bottom aerial photo here, which is from 1958. You can see the plume coming out of a shared drain on the north shore of the river, running right along the north shore. And one of the things that this led to was, if this will switch on me, I don't want to click it a bunch of times because I think it's gonna maybe catch up. Come on, computer. There we go. Is an ongoing issue of elevated concentrations of mercury in the sediments. So prior to about the 1970s, modeling has estimated that annual loads of mercury from these local sources were around 400 kilograms per year. And we can see that in the image on the right. These are spatially weighted surface sediments that were collected over the years, mostly by the federal government, I believe. And downstream of Cornwall, so Cornwall is kind of on the left in this image. This is just the bathymetry and concentrations in Lake St. Francis. The more red the color, the higher the mercury concentration. So downstream of Cornwall, we could see that these elevated concentrations of mercury in the water were being brought downstream with the current and then eventually deposited in the sediments. In the 1970s, a number of industrial processes and regulations came into place, improvements generally that reduced the loads of mercury entering the system. And subsequently, we see a decline downstream from Cornwall of sedimentary mercury concentrations. And we can also see this in our aerial imagery. So if we look at 1972 was actually the year that this effluent outfall was moved from the shoreline to the middle of the river. So you can see it hugging the shoreline on the top, and then later than that, you can't see it anymore in the aerial imagery. And so the big issue that's a concern for a lot of people that was brought up a couple of times this morning is restrictions on fish consumption due to high mercury concentrations in their tissues. Now, this remains an issue in the sediments, mostly just in the Cornwall area, which is what I'm going to be focusing on. So in response to this ongoing contamination along the Cornwall waterfront, the Cornwall sediment strategy was developed, which was designed to essentially assess and remediate these areas. So there were 3 zones that were discovered to have particularly high concentrations of sedimentary mercury. And these were areas that it was thought that sediment was accumulating consistently. So these sediments should be stable. Eventually, if the sediments coming in are clean, that should cap the contaminated sediment. So if we don't disturb it, it should essentially cover up the contaminated sediment and make it not ecologically available. So that was essentially the remediation plan that went into place, monitored natural recovery combined with administrative controls to try to remediate this area. So the problem with mercury, as we see with the fish, is that it biomagnifies up the food web, particularly methylmercury, which is the toxic form. That's going to be our greatest concern. And we know that in the Cornwall area, we have mercury contamination in the sediments. The proportion of mercury that's methylmercury in these sediments is actually quite low. Recently, we found about less than 1%. As we move up the food web, we find still in some areas contaminated invertebrates with elevated mercury, and the proportion of methylmercury gets higher. And as we keep going up into fish, the proportion of methylmercury gets even higher. But we have a bit of a data gap here. What about our primary producers? And I'm particularly interested in the algae. So we know that elevated metal concentrations can inhibit important metabolic processes in algae, things like photosynthesis. It can reduce rates of cell growth and division. And we know that also elevated metal concentrations can cause shifts in algal community structure, reductions in diversity, shifts to what is composing the algae, as well as individual cell deformations known as teratologies. And so the thought is that, well, maybe algae living, on the sediment in these contaminated areas are potentially exposed to elevated mercury and other metals that are contained in the sediments. Now, in terms of what we know about algae and mercury, other metals are reasonably well studied in freshwater systems, particularly those that are commonly seen downstream of mining sites. So we're pretty well versed in the impacts of things like copper and lead and zinc and so on. But and impacts of waterborne mercury have been reasonably well studied both in the lab and in the field. But sedimentary mercury and those impacts on algae are not as well known, particularly at the concentrations that we see in Cornwall, which are quite elevated in some spots. So that leads to our question, which is, do benthic diatoms that are on the sediments respond to this elevated sedimentary metal concentrations, particularly of mercury, since it's so understudied? And has the diatom community structure changed in response to remediation through the Cornwall sediment strategy? So our previous speaker talked a bit about diatoms. They are a group of single-celled algae. They're ubiquitous. You can find them everywhere around the world, in the air, in the soil, on wind currents in Antarctica, and certainly lots of them on the river. And what's important to them about them for us in our lab is that they have a siliceous cell wall. And this means that they preserve well in sediments. The organic part kind of decomposes, but this siliceous wall remains. And these are specific at the species level. So you can look at these diatoms under a microscope, identify them to species. And then some of those taxa have very specific environmental tolerances and optima. So this can allow you to infer what the conditions in the river might have been like at the time when these valves were deposited in the sediments, since oftentimes we don't have those kind of data. In terms of our methods, this was kind of based off of a recent sediment survey that we did, just looking at the mercury concentrations and other contaminants. So we have a subset of the sites that were used in that survey. We have 81 surface sediment sites, the top 2 centimeters of sediment, from our 3 contaminated zones along the Cornwall waterfront, as well as 2 reference areas upstream. We did a number of analyses on these, including all sorts of metals. And we also have 1 sediment core that we analyzed, which is the location is indicated in the blue square in zone 1, which we looked again at some metals and at diatoms. I'm going to be presenting these data in the context of probable effect concentrations, or PECs. If you're familiar with the Ontario Sediment Quality SEL guideline, the severe effect level, this is really similar to that. It's just a set contaminant concentration, it's a constant, above which adverse effects to sediment-dwelling organisms are likely to occur more often than not. So this particular one is based on threshold concentrations from several jurisdictions, including Ontario, and it's unique to each contaminant. And particularly what we're going to be talking about are PEC quotients. So this is just the concentration of your contaminant in the sediment divided by this constant number, your PEC value. So as an example, if we had element X and we found it in the sediment at 3 micrograms per gram dry weight, and we know that its PEC value is 2, our PEC quotient is going to be 1.5, so slightly elevated. And this allows us to easily standardize and compare across contaminants, because any given contaminant is going to have a different PEC value depending on how toxic it is. And again, PECQ values over 1 are likely to indicate adverse effects occurring with sediment-dwelling organisms. We also have a mean PECQ, which is an average across all of our contaminants. So another thing we did was a spatial analysis, a directional spatial analysis. This allows us to essentially determine what proportion of the variation on our diatom assemblage data is attributable to spatial variables, so like spatial autocorrelation. So 2 sites that are really close together are going to be more similar in assemblage simply because they're close together, and maybe the flow of the river could be transporting diatoms from one to the other. This is a directional process though, because we are on a river, so you're not going to get things getting transported upstream. And in this sort of framework, each site is connected to others by edges, and they're given weights to those edges based on the distance between them. So the closer it is, the more of an impact it's going to have. And what you end up with when you do this analysis is a set of spatially structured eigenvectors, which you can use in as predictor variables in multivariate analyses, like a redundancy analysis. So this will allow you to look at, well, how much of the variation in our data is from our spatial structure versus our environmental variables. So in terms of the results, first off, our mean PECQ values here across all of our contaminants, that was formed by 7 different contaminants for which we had PEC values. I don't have them all up here because some of them were not very interesting. The ones that are not presented are all lower than the ones that are. And in terms of the actual results, zones 1 and 2, unsurprisingly, based on what we know about the system, were elevated in both lead and mercury, as well as mean PECQ values. And then zone 2 was also elevated in zinc, which is, again, not surprising just based on the differences in what industries were located where on the river. Zone 3 had the lowest mercury value of the contaminated zones, but is still elevated relative to reference sites. And importantly, most of our PECQ values were below 1. So with the exception of mercury, we shouldn't be seeing a lot of adverse effects being driven by contaminants like lead and zinc. So we have a RDA here. This is the results of our ordination for our surface sediments. We found that we ran these on our environmental data and on our spatial data. Both of those factors influence the assemblage structure in varying amounts. In terms of our diatoms, what we found is that we found several species that tended to co-vary with industrial contamination. They occurred more frequently and were more tolerant of contamination. Then we also found some that were inversely related. So they were very sensitive to this sedimentary pollution. Some of these taxa have been previously recorded in the literature to respond in these ways, and that's great. Others seem to respond opposite to what we would expect. So this might have to do with the bioavailability of the contaminants. The region that we're in on the St. Lawrence River, it's a fairly alkaline, reasonably hard water system. So that might be reducing some of the availability of the contaminants. So when we're looking at what % of our data is attributable to environmental or spatial factors, they're both low but significant amounts, both about 14%. So not a huge amount, but important to note here is that our residual, our unexplained variation is huge, 61%, that's a ton. And this is likely a number of variables that would be influencing the diatom community structure that we didn't measure. So things like light availability, nutrient concentrations, conductivity. So a lot of variation unaccounted for here. In terms of our cell deformations, again, these are mostly in the surface sediments, but there are some in the core as well. on the left-hand side, those two images stacked on the left are normal valves. So on the top is a diatom called Diatoma vulgaris. On the bottom is Tabularia flocculosa. I can't read that, Christina. Okay, 2 minutes. And so if you look at them on the left, that's what it's supposed to look like. So for the top one, you see these nice parallel lines going straight across. On the right, you can see it's like pretty screwed up in some of these cases. On the Tabularia flocculosa on the bottom, more valve deformation, so the outlines are all wonky. And generally, these were moderately correlated with our PEQ values in the surface sediments. And the severity of these deformations was generally mild to moderate, not super severe compared to the literature. Quickly going into the sediment core, the main thing we see here is a shift from a predominantly planktonic assemblage in the bottom of the core to a mostly benthic assemblage in the top of the core. And this seems to occur concurrently with declines in total mercury. But we also have evidence that there were probably also really high nutrients at that point in time. There's been a suggestion that daily BOD loads coming from some of these industries on the North Shore were in the range of 150 to 200 tons per day, which is a huge amount. And again, we can see this coming out in the plume in this aerial image. So it's likely that that was contributing. And so concurrent to the decline in total mercury, we also see a decline in nutrient loads when these regulations went in. So which of these factors is more important? Well, on one hand, like the teratologies are actually pretty highly variable. We have points where we have really high mercury, where we have really low teratologies, and vice versa. So that's not a ton of evidence for the contamination impact. And we also have a lot of eutrophic taxa. These are taxa that really like high nutrient conditions. So these both seem to indicate that probably nutrients were the more important factor. So generally, from our surface sediments, we found that sedimentary contamination, especially of mercury, zinc, and lead, has a small but significant impact on our benthic diatom assemblages, both in the assemblage structure and the incidence of valve deformations. But the natural water chemistry might be playing a protective effect, reducing the bioavailability. Unfortunately, our sediment core resolution is kind of too coarse to really infer how well the remediation has impacted the diatom assemblage. This core was collected in 2016. We might have 1 or 2 intervals in that time between the remediation plan going into place. So it's kind of too recent to really be able to say much. And more generally, paleolimnological investigations in contaminated areas should consider sedimentary contamination as a source of variation because it does have an impact. But also, if you hear hoofbeats, don't think zebras. Other environmental factors like high nutrient concentrations are probably more important. And I think that is it for me. You might have to email me your questions.
Thanks, Katherine. Really interesting presentation. Let me see if there's any. We have like 2 minutes for questions. I'm going to see if there's any in the chat box first. No, but I have one for you. So knowing that you had like a lot of unexplained variation in your surface sediments, do you have any plans, like, I don't know, it's maybe a pain, but you go back and try to measure other factors, or?
Yeah, it would have been nice to do. Really what happened was the River Institute did this survey to look at the mercury concentrations to supplement the data in the nearshore zone. And it was kind of like, oh, well, if you're going out and doing all of this, like, why don't you just give me a little bit of sediment for diatoms? So we ended up expanding the geochemistry a bit to account for that. And so we did like a full metals suite in addition to the mercury analyses. But no, unfortunately, I don't know that it's in the cards.
All right. Let me check if there's another question here. All right, I think that's it. So if you have other questions, you can email Katherine for sure. Her email address is on the website. So thanks, Katherine. We'll move on to the next presentation.